Nothing.
Well, it is noon. We have a little seat swapping happening in the room here. Yeah, no, don't want to cut that off, but you know, we usually get started with some announcements, and as we wait for everybody to be able to get here, and then we'll get going. Not a whole lot on my mind as far as announcements. We're still in the legislative session. And we did get the overall ag budget target, which I think is universally seen as disappointing. So there's not as much money to go around for all things agriculture at the state legislature as we would like, and many of our partners would like. That's too bad, but we still are going to be, you know, responding to requests for information, and our partners are going to be arguing that our bills are still a priority, even with the reduced funding. So I also know that as Jeff Berg mentioned a week ago, the bill for the Clean Water Legacy Act and the Legacy Act overall was heard, and no skeptical questions were raised around Forever Green or other MDA pieces. So seems like that one's moving forward on a good track.
What's the request in that budget?
That's a 6 million for
6
Forever Green. Yep, which is the most we've ever, if it goes through, will be the most we've ever gotten in a biennium. So big opportunities coming from that. Any other announcements, legislative or otherwise?
Well, just to give you another little update. So Mitch and I are just coming back from a meeting with Extension. So if there's any Extension educators online, so we've initiated with Mike Schmidt and the Plant and Soil Science Extension educators initial discussion on what the link should look like between Forever Green and Extension. So a number of you know that we've had links through Extension through the Regional Sustainable Development Partnership with our link with Connie Carlson. We've kept Extension informed, the leadership informed about the Forever Green initiative, and I think we all are now in agreement that there needs to be some sit down conversations and think about how the current members of the Extension program across the state might link with Forever Green, or what some modification of Forever Green Extension positions could be moved forward in the future that would bring those two entities together to support the development of these new crops in partnership with farmers and commercial leaders over the next number of years, basically focusing on the efforts through the pilot studies for the oil seeds. What are the other ones?
For Kernza, the winter camelina, winter barley, and winter rye.
Yeah, so those would be the ones that are being highlighted to initiate that relationship around. So the meeting went, from my vantage point, went well, a lot of interest, but it's a matter of everyone has their day jobs and thinking about how we might make some modifications on both sides. And why don't you just tell them about the person that was just hired?
Well, yeah, we mentioned this a week ago, but we've got a new hire starting. Yeah. Thanks, Luke. We got a new hire starting, I believe, on Monday.
Yes.
April 3rd. Maybe I'm a week off. Starting April 3rd, Matt Levitt from the most recently Albert Lee Seed House. He's been there for quite a while, making great connections across farming communities in the region and really the country. And so he'll be coming on and giving us a lot more capacity to think strategically about, you know, how do we support individual farmers? How do we build programs, potentially in partnership with groups like Extension to move this out in a bigger way?
Yeah, and that's really one of the reasons that the conversation with Extension was initiated was, as we hired people, what is the relationship of these individuals in partnership with Extension? Any other announcements for those of you on Zoom? across the state, across the region. Just one big black hole out there, I guess. Just kidding.
Right, the snow is melting, true, even though it was 17 degrees this morning. when we Got up, so. We're in the freeze thaw, I guess. All right, with that, I think we should turn it over to Brett. Hear about biological nitrogen fixation.
Well, let me say a little bit about Brett. So some of us have been watching his work for a number of years. And with this whole idea, as we work on the winter oilseed crops, it would be fantastic if we can move these oilseed crops into the various rotations without having to apply any nitrogen, right? So as most of you know, there's companies that have developed and are actually putting products into the marketplace basically focused on corn for freestanding nitrogen fixation in the corn system. And the reason I know that is my brother Roger is one of the major investors in that. What's the name of the company? Pivot. And so they have some commercial, or I don't know the details, but commercial product in the marketplace now. So basically it's looking at that general concept to see whether that could be applied to our 4 evergreen crops and specifically the oilseed crops. So, and having Brett here on campus, it's a great opportunity for us with our new crops and his program and his connections to that world to actually, I think, be in a leadership position if we were able to figure out how to tie these 2 efforts together. So with that, you can tell the truth.
Sure. Yeah, thank you for the invitation. I've been here at the university for about 12 years. That being said, there's still quite a few people who don't know me. And so just a little bit of background. I'm in bioproducts and biosystems engineering. My training is mainly as a biochemist, but more these days than anything, I'm probably more of a microbiologist. Just to give you an idea, my lab works on a number of different things. You'll see some of those kind of intertwined here as we go through things. But we're looking at phototrophs, specifically types of algae for production of oil and simple sugars, bacteria for production of oils. We've got a project going on right now on biodegradation of polyethylene, and then what I'll be talking about today, which is the diazotrophic microbes and biofertilizer applications. And then we do some bioreactor stuff, which you're going to see as well in the talk a little bit, because to be able to do the work we do, we have to use certain bioreactors at different times. So just to give you a little bit of history on the evolution of our biofertilizer work that we've done since coming to the University of Minnesota. We started off in this, my lab anyway, thinking about this because when I first got here, as I mentioned before, we were looking at working with algae. And so this was some of the work that I was doing while I was still a research assistant professor at Utah State University, where we were trying to grow large amounts of algae. And as I came here and transitioned, I realized one thing I didn't want to do is try to manage facilities like this outdoors here, just because it's a lot of work to do something like that. But also wanted to still be involved in algae and the potential for it. But we also kind of looked at where we could kind of have a fit. And one of the biggest concerns I was seeing when we were working with some of these people that were trying to do this on a large scale is that the cost still ran into an issue. And that's related again to nitrogen, because one of the benefits that algae was promoted and touted for was this idea that you're going to get significantly higher yields of oils off of a square acre of land. The issue with that is that algae is still a plant, and so algae still needs its sunlight, still needs its water, and it still needs its carbon, but it also needs these macronutrients, so your nitrogen, your phosphorus, your sulfur compounds, to make biomass. And so even if you increase the biomass in terms of the oil, you had to have a similar increase in the total biomass. And if you did that and started looking at algae, what you really were going to end up doing is taking probably about a quarter of any oil you made and ship it right back to a plant to make more nitrogen fertilizers. So there's going to be a huge concern there, and that's one of the reasons why we started saying, well, we really should be thinking about this nitrogen problem in algae. And again, because of where I was coming from, there is an alternative to having to think about using Haber-Bosch process to do industrial nitrogen fixation, and that's the biological nitrogen fixation. And as luck would have it, my 6 years prior to this was working and studying the enzyme here, nitrogenase, that is capable of taking nitrogen and converting it into ammonia. So I already had a pretty good understanding of that, and I already actually done quite a bit of work with microbes that did this process. So I already kind of had a starting point to be able to think about and work on this. And so this was the initial premise that we had for our work. This idea that we would work with certain algae that are capable of fixing carbon and converting that carbon to some degree into sugars and releasing that into the extracellular medium. We work with an organism called Azotobacter vinelandii. It's our primary strain that we've worked in for the last 20 years. And one of the reasons we work on that specifically is because it is an aerobic diazotroph. It is capable of doing this under oxygen. Not only is it capable, it actually gets angry if you take the oxygen away, which is interesting because the enzymes that do this are sensitive to oxygen. So most of the diazotrophs that you hear about are anaerobes or microaerobes. Azotobacter's the opposite. It wants as much oxygen as it can handle. And because you can't really grow algae and harness your azotobacter in a root on a microalgae, this was the organism we thought would be the most beneficial to do this work in. So we started off with azotobacter primarily because of this whole microalgae issue and our concerns there. So one of the things we tried to do is look at ways we could potentially redirect more nitrogen outside of the cell. So we wanted to basically convert this into a fertilizer. We wanted to feed it sugars, and we wanted it to produce different nitrogen compounds. We looked at different compounds, what was known in the literature. So whether we've got ammonia, urea, siderophores, amino acids, and polyglutamate. So those are some of the different compounds. Azotobacter doesn't make polyglutamate. These are just some of the different compounds we were looking at. Azotobacter does produce urea under certain conditions. There's some things known about ammonia. I'll talk a little bit about siderophores here in a moment as well, and does make some amino acids. The other thing we were looking at is, developing some approaches to be able to screen these different microbes, and then also optimizing any nitrogen that we produce. So those were all things we were really wanting to try to do. And the two I've highlighted here are the ones that kind of are associated with our primary project here. We're also looking at understanding all the genes that are responsible for part of this, and the ones that are not just upregulated, but also the ones that are essential. So I'm just going to talk briefly about our siderophore project. It's one of the first places we started with this. And siderophores are, again, siderophores are our compound that bacteria produce specifically to mine metals. So these are things that oftentimes are seen as part of biofertilizer schemes to get more iron or molybdenum or other types of metals that they may need from the environment, and solubilize them and bring them back to the organism. We know that Azotobacter produces huge amounts of siderophores when it's trying to fix nitrogen, because it's trying to scavenge for iron. And so these cultures actually will turn almost green with the color of these siderophores. What these siderophores look like are these types of compounds. So they're either one class is called catechol siderophores. The other is called this polyketide called azotobactin, at least in Azotobacter, that's the name of it. And the key here is that I've circled here all the different nitrogens that are in these molecules. And specifically for the azotobactin, this is a small peptide that gets secreted out into the media with the idea that it comes back to the azotobacter, to the bacterium. And one of the things I thought is, I don't think all of it makes it back. I think a lot of it probably goes and so one of the early projects we looked at was trying to figure out if any microalgae are able to use the siderophore, not for the metals, but actually for the nitrogen. So we grew these together and we're able to show that there are certain algae that are capable of taking these up and using them as their sole nitrogen source. So I'm not really going to talk any more about siderophores, just to give you an idea of one of the early experiments that we did trying to show this. Most of what I will be talking about today, even though we started with urea, is focusing on ammonia, because ammonia is the most direct product, and it's the one that we've been able to drive up to the highest concentrations in these organisms. So that's most of what you're seeing today when we're talking about extracellular nitrogen. We are specifically talking about ammonium, which is just the form it's in when it's in a natural pH or a central pH. So this is another idea of kind of some of the early approaches that we take. And I'm not going to go into too much detail on this and the technical aspects of it. This is all published data. But one of the things we did was designed some strains of azotobacter to serve as biosensors. So what we did is we took some azotobacter, we deleted the ability of these to fix nitrogen, and then we also put some genes in these that would turn them blue if they were growing. And so just a fairly simple technique here. And then we did some genetic screens where we put in transposons, which are just little kind of bacterial viruses, best way to probably describe them, that insert randomly in the genome to try to identify strains that are producing extracellular nitrogen in any form. We show here urea, but this could be any form. And we would be able to sense that by growing this together on the plate with it. What that looks like is this. So here's a plate where we have thousands of colonies, and the colonies that are producing extracellular nitrogen enable the growth of this other strain that's in the background. So you see this blue color show up in the center of the colonies. This is a further experiment we can do where we actually just grow this together with a green algae, and you can see the same kind of effect. So we can use either algae as a biosensor, or we can use these strains that we've developed as biosensors to look for that extracellular nitrogen. And based on that, after we uncovered some of these early genes, we also looked to see how much nitrogen can we get to the plant. So algae is a surrogate for the plant for us. In early work, it wasn't the surrogate. Algae was what we were interested in. And so this is just showing some algae grown together with the azotobacter. This is the wild type azotobacter strain growing. This is the strain that is releasing extracellular nitrogen. So this is one of our early mutations that we found and identified, and kind of demonstrates just what we could achieve. So this is quite a while back. Since then, we have really worked to optimize. So we have done quite a few different manipulations, specifically in azotobacter, to drive as much nitrogen extracellularly as we possibly can. And one of the things that we've been able to do with that is look at exactly how much nitrogen we're getting per sugar. So this experiment was instrumental in showing that we were tracking the sugar loss over time, and we were tracking the amount of ammonia that we're producing. And this sugar here is sucrose. So sucrose is disaccharide. And from 1 molecule of sucrose, we're basically achieving 1 molecule of ammonia. So it gives a pretty good picture of what organisms might be capable of under these situations. And that's kind of our benchmark of what we use. There are some trade-offs. As I mentioned earlier with azotobacter wants the oxygen. So if we actually grow at higher RPMs when we shake, so we're getting more oxygen transport into the liquid solution, we actually can produce ammonia much faster. So it shows that the strain actually was defective in some of the abilities of what it could do based on the amount of oxygen we're seeing transported in there, which again, that's just is a potential benefit for growing together with microalgae, which are going to be producing high concentrations of oxygen. And so it also shows that there's a trade-off. This is cell mass that we're producing. So if we grow with more oxygen, we're making more ammonia, but we're producing less biomass of the organism. So, and this is just kind of a visual. I brought the plate, which I'll hand around to those people who are present so they can kind of see it. This is when we've optimized our organism, kind of what it looks like. The center here, what you're seeing, oops. I don't know why you're not seeing it. Oops, that's not what I want. There we go. The center here is our azotobacter. It's actually kind of a brownish color. That's from the nitrogenase that's being produced. And I've spotted onto the center spot an algae. I've also spotted just algae alone surrounding this. And so 9 more spots here. And you can see, not only are we supporting the algae that's growing right on top of the azotobacter, but we're producing enough nitrogen that you can actually see that nitrogen coming to these algae that are on the periphery of that area. So it's kind of an idea of just seeing just what kind of yields of nitrogen are coming out of this. We're cheating here in the fact that the algae is giving nothing to the bacteria in this case. There's no nitrogen on this plate. There is the azotobacter, but there's a whole lot of sugar already in the plate. So this is growing on a plate with sugar. We also started working with gene essentiality experiments, and those are going to be integral to what I'll be describing in kind of the current directions we're working on in the lab. And I won't go into too much detail into gene essentiality, except to explain what you do is yield a library of tens of thousands of mutants. I think in this library specifically, we figured we had some 60 to 80,000 potentially unique mutants. And those insert into genes randomly. And then if you use next generation sequencing, you can actually look and find out genes that are important to a process. So in this case, what we do is we grow with nitrogen present and then without nitrogen. And strains where these genes disappear from your data are basically telling you that you have a fitness defect, so that those are essential to the process. This is an important proof of concept to run with any experiment, something you know, because what we did in these conditions is we were looking at the genes that are actually part of the nitrogen fixation. For nitrogen fixation, the primary genes that do that are the HDK NIF genes. And you can see up here that we have the NIF-H, K, and D genes that all showed significant defects when we tried to grow them without nitrogen if those genes are disrupted. So that's just kind of a initial piece of it. I have some other diagrams that kind of show what this looks like, but I'm not going to go into the technical details of that except to explain that it's a tool for our research. So around 2014 and 2016, we sat down and thought, all right, we're doing all this stuff in algae. I wonder if any of this would translate to current agriculture. So we actually put together a project that was funded by MinDrive. It's a transdisciplinary research program. We were working together at the time with Craig Schieffer and started working with Neil Olszewski as well, trying to see just what we could do. And we went for gold on this. We said, okay, we're going to try to see what we can do by putting our azotobacter together in the soil with corn and wheat. And we started doing some of those experiments and really did not see the types of effects that we see in algae. We kind of threw the kitchen sink at this. We tried doing things with hydroponics. We tried doing all kinds of things with killing off the other microbes in the soil. And we even just took liquid media that was spent from azotobacter cultures and tried just putting that onto our plants to see if we could see any kind of increase. And we really weren't seeing it. We were seeing some slight things, but the air and the noise and that really wasn't showing and demonstrating.
What soil was used? Was it?
We have to ask Craig for that. Yeah. So we played around with it. We got the soil out of the equation by going to hydroponics and playing with that.
And still didn't see it.
Well, contaminant. We went right to the greenhouse. We probably skipped over some primary steps that we should have done earlier. And so, coming out of the project, I don't want to say it was a failure because it wasn't. We did quite a few different things and we learned quite a bit as a result, but we didn't really see the yields we wanted to see. And so we did a little bit of reflection afterwards and tried to do some regrouping and thought about what was working before and what didn't work when we went into the plant system. So first question was like, well, are we making ammonia? I showed you the plates, we showed you the algae stuff. We're definitely making ammonia. We can quantify it. So we know we're making nitrogen. We know that azotobacter species are often used in plant growth promoting biofertilizer applications. Get broadly used in parts of Asia and parts of India. And even here, some people will put inoculums in that have azotobacter. Whether that's for nitrogen or whether that's for siderophores or something else, or phosphate solubilization or potassium solubilization.
That's the species BioPivot uses.
That's what Pivot is licensed from us, yes.
Yeah.
So we've done this.
But I mean, the commercial one.
I don't think that's what they're using commercially now. I get all of this secondhand, so I don't have a direct connection to Pivot specifically. I think it's working with certain crops. So it is still showing some promise with certain crops. In our hands, we weren't super convinced by that, but we were doing some small scale greenhouse stuff. I want to leave the large scale field studies to people who can handle that. So I'm happy that they're licensing that technology and trying to utilize it. I don't know that they're necessarily using our strain just because they don't tell us that.
So you're saying even in the hydroponic system, it didn't promote extra growth?
We ran into contamination issues. We didn't really see it, and I'll kind of get into why we didn't see it, because we've got data now that kind of starts telling us a little bit of that. For the record, we've not given up on azotobacter. This ability to fix nitrogen aerobically has other potential. So while we say we've made some changes now, I have not given up on azotobacter. It's still my organism of choice. This is just a recent literature article talking about all the benefits of azotobacter. And it shows here, talking about this as a potential biological enhancers for crop nutrition. And it shows these as free living nitrogen fixing bacteria, but it kind of shows these fairly closely associated with the root. The truth is this is an aerobe. So it's probably not down here very far because it can't do anything anaerobically. These are probably growing closer to over here and probably a reasonable distance away. You find them in soils, you don't tend to find them overly enriched in the rhizosphere. So other people are doing this, and this is something that's been in the literature for decades, that azotobacter is associated with plants, but I don't know that it's necessarily this tightly associated. So as we stepped back and started talking about this and what we maybe would try to do next, we thought about what we really want. We want a microbe that we know works well as a good plant model, something that is demonstrated to be found associated with plants routinely. We want broader application. There's tons of people studying root nodules, and there's no reason for us to jump into that arena. There's plenty of people working in there. So we actually wanted something that was found in plants, but not limited to that. We wanted it to be something that might be applicable to other crops. Wheat specifically was one of the big ones we were going after, but even corn would be nice to at some point be working on corn as well. It needed to be nitrogen fixing. We still wanted to be able to provide additional nitrogen. And we wanted lower competition with other soil microbes. So we didn't want to be fighting with the other microbes or having that nitrogen being fixed and then converted into other forms of nitrogen that the plant never sees. And then the last thing we really wanted was some specificity. So what we thought about after we went through this is it's probably more important that we think about the location of the microbes. And we started moving into microbes that are known endophytes. And an endophyte is a microbe that is found associated with the plant in a way that it's protected from bleach, basically. That's kind of the definition used for endophytes. So they can be in the root, but specifically, you can find endophytes in other parts of the plant. This is one of the reasons why I haven't given up on Azotobacter, because Azotobacter could probably function well in the leaf if it didn't make the plant sick if you put it into the plant. So we really wanted to think about things that were true endophyte bacteria. And that's where most of our work has been moving towards, is getting more in touch with some of these endophyte microbes and starting to work with them. So the organism we switched over to is this Gluconacidobacter diazotropicus. There are some differences between these two microbes. The Azotobacter is an aerobic diazotrope. Again, it's categorized as free living because it's not generally associated with plants in the rhizosphere, whereas Gluconacidobacter diazotropicus is a microaerobic endophyte microbe, and it was originally identified from an association with sugarcane. So that's where it's come from.
So that sugar cane association is real.
That's where they find it. You
Understand?
find it in sugarcane stalks, and in Brazil, people inoculate crops routinely with Gluconacidobacter diazotropicus.
Yeah, I remember that big debate.
Yeah.
Over,
Yeah. So some of the benefits, it is able to fix atmospheric nitrogen through nitrogenase. It's still debated how much nitrogen Gluconacidobacter fixes in sugar beets or sugarcane, right? It's reported to form endophyte associations with a broader range of plants. And again, these are non-nodular associations. So people have reported it to form associations with wheat, form associations with Arabidopsis. It's got a smaller genome than Azotobacter. It's actually a less complex organism in many ways. And it's microaerobic, meaning that it potentially could grow in a different range of the plant, specifically in the roots, but also in other tissues of the plant. So those were the reasons that we selected Gluconacidobacter. It's not the only one we selected, but it was our primary target. And so that gets us to our current USDA project, which has been funded since 2020, although we are nearing the end of the stage on this project. So we are actively looking for additional funding. Again, I'm going to kind of come back to this. This project is really dependent on these libraries where we use transposon sequencing, where we generate huge libraries of mutants, and then through next generation sequencing, use that to tell us who's important, what genes are important in something. Basically, as long as the gene is not essential for just life on a rich medium, which there are some genes you cannot delete, but there's a whole large amount of the genes in the organism that are only really important for certain conditions. And that's kind of what we wanted to do. One of the things we want to do is I understand specifically the genes that are important to forming the endophyte relationship. So we'd really like to have a clear picture of what that is, what governs that endophyte relationship, right? So that's one of the reasons we've gone through this. You'll see it's actually generated some other pieces of working towards that approach, as far as other things we're able to study as well.
Does it go the other way too, that you're looking at things that get knocked out that improve?
Yes. Yes. So I don't have too much of that in here because that data is still preliminary enough that I'm not comfortable sharing it, and we've got to get some IP on some of that stuff. But definitely, even in some of the work we've done and published, we have seen genes just for nitrogen fixation that when they've been deleted, improved nitrogen fixation. I want to be cautious there because when we fix nitrogen in Gluconacidobacter microaerobically, we do it in a condition that looks nothing like the plant. We are doing it in a liquid culture that we're keeping the atmosphere at a microaerobic condition. So some of that data may be important, some of it may not as far as for nitrogen fixation. It might have to do more with the culture conditions. All right, so this is the first objective. I'm not gonna go through every objective of what we're working on this project. I just want to kind of give you an overview. So our first objective was to employ whole genome gene essentiality screening, this TNC, with this established Gluconacetabacter diazotrophicus. And then to do that while we're growing it in either Arabidopsis or wheat. The Arabidopsis is just our model plant system that we wanted to focus on, and then we wanted to also suggest we'd work with an actual agricultural crop that is grown in large scale. So we wanted to do several different things as part of this objective: construct TNC libraries of Gluconacetabacter diazotrophicus and Azotobacter vinelandii. We have published some papers on that already, specifically just related to nitrogen fixation. We wanted to test the TNC library independent of plants, which again we published, to determine genes that were essential to the organism as a baseline. So things that you can't get rid of no matter what without killing the organism off, and to establish a baseline before we go into plants. And so we wanted to look at that. As I mentioned, we already did that just for nitrogen fixation and liquid culture. Then the final thing we really wanted to do was getting at this: what are the genes that are essential to establishing this endophyte relationship? So we wanted to grow this library together with our target plant species and determine the genes that are essential to the endophyte relationship itself. So this is just kind of a visual of kind of the idea behind it, where we take this large library. Our Gluconacetabacter library, we believe, is about 80,000 unique mutations that we introduce and then grow together. I don't know where my arrow keeps going. We grow together with our plant. And then from that, we then isolate the bacteria back. And so one issue with this is you have to make sure that you get good infection. So you have to make sure that you're getting lots and lots of bacteria into the plant. And so we've gone in the literature to see what we believe governs that, because to get the bacteria into the plant, we basically mix the seed together with the bacterium, and then use that to inoculate and put the seeds in. And so this was the original idea of how this would look, is that we would have to sit there and grow several different times with plants to enrich for these. The truth is we're actually getting really good data after just one try. So when we actually go through and work with this, we're getting what we want. This is where we get into the problem, which is where we kind of ended up working with some evergreen crops. So this is the different seeds for some of the different things that we're focused on. I really don't know where there's my arrow. So here you can see wheat seeds next to our dime. We've got some tomato seeds, and down here is Arabidopsis. And this was the one problem we were getting into. We're trying to add 80,000 mutations into a seed to then propagate inside of that, and then get enough plant biomass to isolate the bacteria back from that and get a significant enough mass of bacteria to actually determine genes that are essential. And so Neil, and talking with David Marks, sat and said, well, maybe we should switch away from Arabidopsis over to pennycress. I believe that's my pennycress seed. Right.
First time anyone has said that pennycress seed size is ideal.
Well, yeah. And if I had known that pennycress is also a relative of canola, I might have pushed for canola.
Oh,
But I didn't know that, and so
thank
we're
God,
actually,
thank God there's some ignorance.
we're at, well, this is why I'm on board. Right? So we needed sufficient root mass to get enough mutants from it. So we made this shift from Arabidopsis to pennycress. Again, our reasoning for that was purely selfish, because we knew it would work better than the Arabidopsis was working as far as growing plants. We are still working on wheat, and in fact, most of the plant studies we're doing right now are wheat. We do pennycress. I'm going to show you what we do pennycress with. This is the apparatus that we use to grow things, and Neil's done most of the work as far as developing this in his lab with his students. Calls this system the ragdoll system. It's really not soil. So the ragdoll is basically a paper membrane a paper and a membrane that's kind of wrapped around. You can see down in the bottom of this, we have some liquid media that has, I think little to no nitrogen in it. So it's very low. And then the seeds, after we mix them together with our gluconacetobacter, we go ahead and plant these and we grow them until we get sufficient enough biomass to do that. The ragdoll is important because it's really important that we get a root structure that is amenable to infection, and that is important. You've got to get these lateral roots that form, because that's where the bacteria are believed to actually infect the plant. And one nice thing about this is when you're done, you can very easily separate the roots without having a whole bunch of soil and other things getting in your way. It's a lot easier to keep sterile and not have to be contaminated. The whole apparatus can be autoclaved prior to adding the seeds. So we really are starting off with a fairly pristine environment for the seeds. In the process of doing all this, as we started working with the pennycress, we started realizing we could actually grow these seedlings just in flasks. So if we could get the sterility issues taken care of, we could grow the pennycress together with our gluconacetobacter and actually see the gluconacetobacter growing in there. The benefit of this is we are not growing the gluconacetobacter on sugar. We're growing it on what the seedling is providing. So this is the experiment that was a side experiment that came out of all this. That's a lot faster to do, but it starts telling us a little bit about the genes that are important to utilizing root exudates, what the plant is giving the bacteria to benefit the bacteria. Cool. So a fairly simple experiment to do. I say that because I don't ever do it. People that actually do it, every so often these will get contaminated or they completely fail, but it's fairly reproducible and it gets us a pretty high yield in a fairly short amount of time. So we did quite a bit, and I say we, I mean Neil's folks did quite a bit on optimizing the plant side of things. So finding a suitable library size to sample a majority of genes, proper methods to sterilize the seeds, which we still play with and have to work with as we switch to different seeds. Required number of plants to get suitable number of infections in the ragdoll apparatus, sterilization of the apparatus, harvesting the bacteria in isolation, and then the sequencing turnaround is usually 2 to 3 months to get the data back once we do an experiment. But this is what the results start to look like for us. And again, you don't need to know too much about TNC, except it's a fitness thing. So most genes will have a fitness, and it's not even a fitness here because we're not calculating it. It's what we call fitness. It's the fraction that still remains from what we started with, our initial population. It's the number of times we see that same number of genes for that specific mutation. So anything that's in blue right around 1 didn't really change as we did the experiment. Anything that decreased, that mutation was bad. That mutation was essential to growing. In this case, we're growing in the presence of the roots. So we're growing just in these shake flasks. And it gives you an idea of just how reproducible the results are. We have mutations that are very, very important. I've only shown you a selection of them, just because we haven't published this yet. But just to give you some idea of what the reproducibility looks like. And then to also show you genes in close proximity. So this 726 is really close to 724 in the genome. 724 didn't care at all. Go ahead and mutate me. But 726, I'm not going to be happy growing with plants anymore. So we also narrowed it down to a very small set of genes that seem to be essential to growing on root exudates. And it starts telling us what's important about forming this association. Again, not in the plant, but just growing in the location around the plant. There is a flip side.
But
David,
you don't know whether it's in though either, right?
in this
No.
situation, we know it's not, because we're actually only taking the cells from outside of the plant, the stuff that's still growing.
Right.
We also are starting to transition to the ones that are inside in those experiments as well. But there we can't say which ones are just on the surface of the plant and which ones are internal. We really have to do the other experiment that we show you. The flip side here that I want to mention is what David's already pointed out. We do see some genes that deleting them improve our growth. So they show up at a higher population. In these experiments, we haven't seen ones that jump drastically, but we're starting to see some drastic ones when we get into the actual ragdoll experiments. So that's all I'm going to talk about. Like I said, I'm not going to show the ragdoll data just because we only have one full data set. It took us quite a bit of time to figure out all the conditions to get that to run really well. We did an initial set just to see if we had enough mutants from the scale that we were running, to realize we had enough mutants, but we really needed about 4 times more to get a really good data set. We've got that data set now. We're just starting to work through that. And that's with wheat. This is for pennycress specifically that I'm showing you right now. So that was our first objective. The other side of this is we're trying to understand the genes that are important to the endophyte, but we are also trying to take the endophyte and make it a better nitrogen fixer. Okay, so the other part of the objective here was to construct high nitrogen yielding strains of our endophyte. That was based on either disrupting the genes that are important to keeping the nitrogen inside the cell, and also some disruptions that we do to deregulate the organism so that it produces a lot more nitrogen. And then also develop some methods. Because this is a microaerobic type of setup, we have had to do some things in the laboratory to really demonstrate this. The other thing I want to point out here is all of our modifications that we're making in this, we're thinking in terms of agriculture. So we are not doing transgenics for this. We are doing some transgenic work in the laboratory where we add other genes, but this is just gene deletions or segments of genes that we're deleting. So nothing's foreign's being added to any of the plants, just so that we can be thinking in terms of potential regulatory aspects of this in the future. So
Is this being done with CRISPR then, or what are you?
no, we use a selection counter selection. I'll show it briefly. I won't go through it in detail. It's just a way of adding things and then removing them afterwards through a counter selection technique. So everything CRISPR can do, we can do. I don't know if it's quite as sleek as what CRISPR does in some cases, but it works and it's effective, and we get manipulations here. This is just the schematic kind of showing this. So any of the mutations that we're doing, here's one of the mutations we targeted, which is a transporter, specifically that's supposed to keep any nitrogen the cell makes inside the cell. And so what we've done is we've targeted for deletion. We have a 2-step process we go through. Actually, it's 3 steps to actually isolate them when we're fully done, where we first of all delete the gene and replace it with a marker. So it does have an antibiotic during this step. Then we do another selection technique where we insert things in such a way that we put a toxic gene in place. And then following all of this, we get 2 different options for how these can kind of recombine. But when we're done, as you see, this is that segment of the genome and what it looked like beforehand. This is the final product. Nothing is foreign remaining. All we've done is taken that gene completely out of the genome. So it has no antibiotic resistances. It's just missing that gene altogether. So that's the approach that we use for making this.
What's the name of that technique? What's
It's just called a selection counter selection. You use a gene that is conditionally toxic.
Yeah.
And there's different ones we've used in some of our other papers. In this case, it's actually sucrose. So you can go out on glucose or sucrose. If SAC-B is present, it makes a polymer that builds up inside the cell and basically causes the cells to goop up inside. So they can't get rid of that. We also had to do a little bit of work to be able to actually test and see that we're producing additional nitrogen. Gluconoacetobacter is a diazotroph, and trying to measure especially small amounts of nitrogen. We still like using our screens. We still like using these biosensors. So this apparatus just basically has an atmosphere that's being introduced to it of this microaerobic. So we do it about 2 to 5% oxygen that we pump in. The remaining stuff comes back out. This is just to make sure that the headspace inside here stays constant. So it's just to make sure you don't have oxygen leaking back into the system. So we flush this atmospheric mixture through. You also can see we've surrounded this with a LED system so that we can grow our algae, which is our biosensor in this case, with the gluconacetabacter. And so that's the system that we have in the laboratory, so we can screen for different things. This is kind of what the results look like. I already showed you the one plate with the Azotobacter. This is now gluconacetabacter. Here's the wild type. Here's the wild type with some manipulations to overexpress nitrogenase. Here's the wild type with a single deletion of one of our transporters. And then this is the double transporter deletion. And this is a combination of 3 different manipulations that we do to get to this strain right here. This is early on in the growth, so you can see slight improvements here for these ones individually, increased improvements for combinations, and you really can't see a huge difference here between these two because it's getting enough nitrogen under these conditions that you just can't see it with this approach. We have to go to a different approach, which I thought I'd maybe show you a video of. It did not work going from my iPhone to my HP, so I haven't got the video up here. This is showing what these look like if you let them grow for a couple of weeks. So you can see just how much you can yield in terms of algal biomass. The ammonium levels, we have to actually measure this with liquid culture. So we do have some culture solutions or systems that we use to do that, where we grow these in flasks, and we grow them initially in flasks or initially in an apparatus that's a turbidostat. It's hard to explain that without showing it to you, but it allows us to grow them microaerobically initially. And then when we actually try to see how much nitrogen we produce, there's a point where the cell density gets high enough, you can actually pull the organism out of the microaerobic conditions because it starts behaving like Azotobacter. Now it wants more oxygen to produce more nitrogen. So wild type, the quantities of nitrogen we're seeing are in the micromolar range. Our levels of ammonium that we're getting with our modified strain is reaching up as high as 4 millimolar before the culture seems to crash. Probably either changing the pH, gluconacetabacter actually acidifies its media a little bit, or it probably is running out of carbon compounds. So at that point,
Is that high? That 4?
Micromolar is probably around 3 or 4, so it's 1,000-fold increase. So it's doing essentially what we wanted to do, what we did at the Azotobacter, where we put it on steroids for producing nitrogen. But again, just by deleting genes or deleting segments of genes. So we do want to start working in plants in greenhouse conditions. This was something we had proposed early on. These are some Arabidopsis mutants that are highly susceptible to lower nitrogen. So if there's nitrogen minimal in the soil, you don't get as good a growth with them. This was something we had in the project, is something we could potentially use to screen. The screens I've shown you with algae are much faster, so they're quicker to get us to there. And we probably won't get to the greenhouse studies in this grant just because we didn't budget for doing greenhouse work with the project. We weren't certain what things would work and which wouldn't. So this is kind of some of the directions we're thinking about going. Our accomplishments to date, we've completed a full experiment with gluconacetabacter in one time in the ragdolls with a nice data set. We need to do some replicates there. We developed a second method that allows us to actually start studying the genes that are important to root exudates. That's using pennycress. And I just showed you, we constructed this modified strain of gluconacetabacter that has a significant increase in the ammonium when grown alone. And that's not with any plant. It's also not with plant provided exudates or with plant material. It grows in sugarcane. If you can imagine, sugarcane environments probably fairly high in sucrose where these are growing. So it might be a reasonable model for what could happen in sugarcane, but we do not know anything about how well this will translate in plants. We're playing with that in the lab. We will not be able to get through that with the amount of time and the money we have left over in this proposals or in this grant project as it stands now. Some of the future work is determining if we can see a measurable improvement in the plants. We're playing, like I say, with pennycress, wheat, and a couple other plants right now that we're doing. Determining if genes that enable growth of root exudates might differ between different crops. So we can just simply drop other seeds in there and see if the same genes are important for every crop or if they differ when we go to different crops. Investigating genes that result in an improved growth in our endophytes. And then running similar experiments with other bacteria. So on my side of things, we have created generated the organism that produces the high nitrogen for this organism. We'll probably be waiting for quite a while till we've got definitive data on that, but we'd like to look at other endophytes as well. And as I said, we didn't just start with Gluconacidobacter, it's the one we focused our attention on. So we have some other endophytes we'd like to look at as well. Modifying additional endophyte bacteria for nitrogen production, identifying the target genes to target. Let's see what I'm trying to say here. Identify additional target crops and target endophytes, and then again trying to get some funding to do some of the greenhouse type studies to move to the next step. These are some of the people that worked in my lab. As I said, we've worked with Neil for the USDA project, previously worked with Craig Schiefer, and some of the different folks that have been doing a lot of this work and working through all these obstacles to get us to where we're at. And I think I did it in close to 45 minutes. So,
Well, we usually go, as you well know.
Yeah.
We don't have an exact stop time, but really appreciate that grand overview. And as we will, as you know, and all of us know, Craig Schaefer and Jake Jungers, they've been tied to the Forever Green Initiative from the very beginning. And Jake certainly has strong interest in this work, as well as others. So we're very interested to figure out how to maintain that link of your program to the forever green crops and beyond. So it is that whole area, as we all know, going from the laboratory into that hellhole with soil. And that's what I was just thinking, that I think we would have a core of folks, Neil and others, that would be very interesting to think about, the evaluation that does have a closer and closer tie, to that soil environment. So just rest assured, there is a core that would be very interested in helping move that closer and closer, whatever the hell that is, of getting closer and closer to that environment.
a big piece of going to Gluconacidobacter is actually to remove soil from the equation to a certain degree.
I understand, yeah.
If we get into the roots and we're in as an endophyte, we don't have to worry about some of those competitions. Hopefully the plant has first access to any nitrogen we make. So, and we have a more direct link potentially to carbon sources that might be.
But as you well know, there might very well be that microbe that will reach in and kill it.
I know. Yes, we do know. We do know this.
We do know this.
Yeah.
Do you think that either one of these two main microbes could function as an endophyte in the stems or leaves and fix nitrogen in those?
There's arguments for where Gluconacidobacter does reside, whether it's predominantly in roots. In sugarcane, they find it in above ground tissues,
Including leaves. Or stem, is it stem?
What people report and what we have done are different things.
Yeah.
So Neil does have an experiment and talk about here, but one I'm really excited about, where he basically forces the bacteria into the leaves, and then re-isolates the bacteria from the leaves over time to see who really shows up. And Gluconacidobacter doesn't seem to proliferate in the leaves, but it doesn't go away. So it stays there. It doesn't grow out. The other endophytes that we're interested in go into the leaves and propagate within the leaves.
Let's open it up for questions from folks on Zoom.
Yep, Ford Dennison has his hand up. Go ahead, Ford.
Um, so, you inoculate your plant with your strain, and pretty soon we have a plant that has that strain plus its various mutants in the plant. And so the question is, if you have a mutant that puts less energy into fixing nitrogen for the plant, is that mutant then going to reproduce faster in the plant, and you end up basically with your plant dominated by non-fixing or less fixing mutants?
So we've done the experiment once to know that there are ones that, I don't say they dominate, they're increasing. We introduce everybody into these, we do multiple plants, and we hit them all with the same library, basically. So only some of those organisms from that library get into the root. And so we expect that we're going to find ones that decrease because they didn't get into the root, because certain genes are essential to that initial infection, is what we call that piece of it. The ones that have gone up do not seem to be producing less nitrogen, seem to be producing or less of, well, they're disruptions in other genes. And we don't know exactly what all those genes do. We're playing with some of those, but it's still a little preliminary for us, other than to say that it hasn't been an effect yet where something came over and dominated the plant in that one.
Well, just a simple experiment between your wild type and your high fixation one would be an interesting experiment.
So that is being done. We're playing with those experiments to see if one outcompetes the other. So, and we're also looking to see if the ones that are showing up in high, that are increasing in the plant, if those outcompete the wild type as well, or if there's some other artifact that we're missing. But so far, we haven't gotten to that. It takes us about a month to run the experiment, to grow up the plants, and they don't always grow. Every so often something happens where the seedlings just don't germinate well in these, and sometimes they work beautifully, and sometimes they work beautifully when you didn't scale up, and as soon as you go to scale up, everything crashes. So it's still, we're still tweaking with things, but no, we haven't seen something where something dominates and ruins the data set. The data sets still look really nice, actually pretty good. I was hoping to see at least 50% of the genes covered, and I think we've got probably close to about 90% of the genes that we're able to get in liquid culture coming back in each data set.
So these are endophytes that are just simply in between the cells of the cell wall.
Neil has some images that we've also done. I mean, other people have done this with Gluconacetabacter, where they put fluorescent labels onto the organisms and show. He's played with that, so he's got a red fluorescent protein looking at roots. I keep pushing Neil to look at things above ground as well, the stems and that, to just see where they're going. But we know that the vast majority of the organism is in the root. It's in the root system.
But he doesn't know whether, yeah.
Well, yeah, you got to run these for long enough, and then you got to get down to the microscopy. And to be honest with you, we're not. I think Neil's more of a microscopist than I am, but we don't do a lot of that, and that wasn't part of our goal for this, because there are papers published on Gluconacetabacter where it resides. Yeah.
Anyone else online?
Yeah, Nick Jordan has a question.
Yeah.
Brett, hi, Nick Jordan from Agronomy and Plant Genetics. Thank you for a really fascinating talk. My question is kind of follows on Ford's, I think. So it seems like this is a spectacular functional alteration, this 1,000-fold increase in ammonium export. And, raising the question of fitness consequences of that functional alteration. And I'm just wondering how large that looms in your proof of concept research agenda. Ford's experiment seems like an obviously good idea, but are there other ways that one might, explore that potential fitness cost of this particular functional alteration. And I'm just wondering how you think of that.
So we've already published a paper on Gluconacetabacter fitness in liquid culture. Okay, where we grew it side by side with nitrogen and without, and looked to see some of the genes we targeted to make sure that we weren't going to dramatically lower the health. And because some of the genes, so Gluconacetabacter ends up having a little bit of gene duplication where the one gene for transport, we had to knock out 2 different copies of it. So in that case, each of those showed a slight fitness defect, but slight, I mean, maybe a 15-20% defect. If you take out both of them, is it a higher defect? We didn't test that in our system. But we grow this side by side against wild type with not just the deletion or not just the genes that make it so that the ammonia gets exported, but also the ones that over regulate and turn up nitrogen fixation. And they're not a big difference in the growth of them. I thought it'd be significantly higher. We're measuring this with the turbidostat, so we're growing them both and looking at how quickly they're replicating, because we're just basically catching them in exponential phase of growth at an optical density of about 0.4 or 0.5. And we don't really see a huge decrease in the growth of our strain that really should be decreased because it's cranking out a lot. Azotobacter only produces about 6 micromolar of ammonia in the extracellular media without no manipulations. It produces up to 30 millimolar. So I mean, it's a sizable increase in what you can generate. It's prone to cheaters. So it is prone to organisms that if you do that, they'll be bathing in so much nitrogen, they'll actually make mutations in the nitrogen fixation genes. And so that's something we work around. And actually deleting the transporters means they can't retake it up very easily either. So so anybody else in that population that you're adding would be at a struggle. So that's going to be a question. But so far, even if we could increase the nitrogen by 5-fold, which we're well above, we expect to see something in the plant. I say that, but I shouldn't, because we haven't done enough stuff in the plants yet to see what happens and how it translates.
We can say it until it isn't true, right?
Yeah. So yeah, we're interested in the fitness, but the fitness is actually, I've been surprised at how little of an effect that had on fitness. I've been more surprised by genes we're finding that seem to be beneficial to the plant or to the microbe when it grows together with the plant. That's what we're kind of more investigating right now is, are there other things that will improve the ability of our strain to outcompete other organisms in the plant?
Well, that's all fascinating. Thanks a lot, Brett.
Are these cold sensitive or cold resistant? Do they stay in the environment or?
If
Yeah,
they're
they
on,
inoculate every
if they're
year.
on a root of a winter annual, will they survive what's out there today?
We don't expect anything we're doing to persist in the environment because they do have a growth defect. While I say it's surprisingly lower than I expected it to be, they're still defective. So we expect wild type to outcompete. I don't know how geographically dispersed Gluconacetabacter is in every soil. This came from the strain we're using, came from Brazil. So it's a sugarcane crop organism. What they do in Brazil is they seed coat. So they have got things that they either spray on the seeds or spray on the plants early on during germination to inoculate. And we haven't worked out what that would look like, but I'm expecting that to translate this, that's essentially what you would do is seed them at the point with your bacterium at the same time as you put them in the ground.
But that's the fall.
Yeah.
In the case of the winter annual, the question is, would it survive that association during, on living roots, right, during the 28-degree temperatures that you have in the soil? Right?
Yeah, and that's something well outside the scope of what we're working on right now. So a good question, but
Yeah, but I mean, those are the type of things we'd be very happy to help think through with you.
so we, even Neil and I have had this discussion. Neither of us are really geared up to do greenhouse studies. If we did that, we would need somebody who does that more often than, does that routinely.
We have
to-
those people.
To be doing that. So we would want to have collaborators that do this
Look at a hand go up.
instead of doing this ourselves. Want to be involved, but really, I'd rather give that piece of the project to somebody else to figure out and figuring out how to optimize, because I'd like to be looking at these other genes that are showing up in our sterile conditions where we can really look at that. I don't know how well gluconacetabacter is going to outcompete other endophytes inside the plant.
I understand the basic nature.
Yeah, so it's there are things there that we'd love to explore. I don't want to just focus on gluconacetabacter for the rest of my life either. I'd rather kind of see what other opportunities there are for that.
Your connection to Craig Schaefer was right at that time, right? So
Yeah.
Craig, as you probably know, is going to be phasing out through retirement. So we'd be very happy to work with you to actually put this new team together, right? So you guys could think through the relationship that we would be able to help
Yeah.
identify some of those folks in soil science and agronomy, plant genetics.
I know this isn't your realm of things, and it's probably not kosher anymore, but it seems to me, I mean, the whole big rigmarole of making a nodule is to make something that's so the rhizobium in an anaerobic conditions. But you identified these nitrogenases that are oxygen insensitive.
No, the organism is insensitive. The nitrogenase, if you pull it out of the organism, is just as sensitive to oxygen.
Okay.
The organism creates an environment. Azotobacter, if you go back into the really old literature, and you're going back to Burke, so the media we grow on is Burke, so that's Lineweaver Burke. So they used to study Azotobacter because the respiratory machinery of this is the highest oxygen consuming organism known. And that was at the time, at least, that was the thing. It's far higher than any other known heterotrophic organism for consuming oxygen. So we actually can see this. There are certain proteins that we put in, like green fluorescent protein, that requires oxygen to make the pigment. Azotobacter doesn't like to make it. It doesn't, there's that little oxygen inside the cell. So it's got a really robust system there. The other interesting thing we have learned with gluconacetabacter is gluconacetabacter, you cannot start it off unless you grow it microaerobically. But when we grow it in turbidostats, we actually have to raise the oxygen as the density gets higher. Otherwise, we don't get it up to a high density. And after it's gotten to that density, we can just take the effluent coming out of the turbidostat, put it in a shake flask and grow it like Azotobacter. So the 4 millimolar that we're getting, that's in a shake flask. It's fully aerobic. Yeah, it's got enough mass that it's doing the same things that Azotobacter does. It will be one of the things in our paper that we'll be mentioning is that it's microaerobic when it needs to be, and it needs that to get over that first hump. But once it's over that, it's a fully aerobic organism and probably has the same types of features protecting that nitrogenase that Azotobacter has. So it's going to be a little different from what's out there in the result or out there in the literature.
Any other questions?
Not from online that I see. Anyone online want to ask a question? Oh, Tim Olmosov raised his hand. I see that now. Go ahead, Tim.
Yeah, thank you. Well, great research, Brett and Neil. And my question is, about sort of how ready you are. You think potentially this invention is for commercialization in terms of is it the right time to start a company, or you're waiting for a specific milestone? And obviously, the main parallel you can draw to Pivot Bio that Don mentioned early on. And I know that Pivot Bio pulled in about $400 million in the investor money, suggesting that there is strong appetite for technology like that in the venture community. And just curious, what are your thoughts about starting a company that will try to commercialize another microbial source of nitrogen based on the discoveries that you made?
So we have IP file on some of the stuff we've done already. It's different because with different organisms, so we're doing different things in that regard as well. Until I see something concrete in a plant system, it's nice. It's cool to see that you can do it in the laboratory. It's teaching us a lot. I see a potential for multiple places where these can be benefits. If we can understand really what governs the endophyte relationship, not all endophytes are beneficial. I didn't point this out. Gluconacetabacter is not a pathogen. That's one of the key things for my research. We don't work with pathogens, but think of all the things that are pathogens that are also endophytes. And so understanding that could be that you now know what genes to target to make it harder for other bacteria to survive as endophytes. And so understanding the endophytes, understanding the genes that are essential to root exudates. It's amazing when you start reading through the root exudate literature, how poorly characterized root exudates are. And we found some evidence in there for maybe why Azotobacter is struggling when we try to grow it with plants. It's just related to the root exudate. It's not sucrose, which is what we grow Azotobacter and Gluconacetabacter on in the lab. So I think there's a lot of other things to be discovered. I definitely would like to see this applied. My goal is to try to limit some of the nitrogen or some of the environmental costs associated with producing fertilizer, but also to try to lower the costs for farmers in fertilizer costs. In the last couple of years, it's been very detrimental for farming as far as the cost of fertilizer being so out of hand. And so it's not just an economic cost, but also the environmental costs of it. And, if you think about it, decades ago, we weren't putting all this nitrogen on our soils, and we still were producing crops, nowhere near what we produce today, but we were producing crops. So biological nitrogen fixation used to present 80–90%, the rest coming from lightning strikes. But now it's producing about 60% of the nitrogen out there.
So in the United States, how many labs are there organized like yours, looking at the nitrogen fixation issue, freestanding nitrogen fixation issue for incorporation in cropping systems, right? Who's doing? How many labs are doing the basic research that you're doing?
So if you start looking at the people that are going for the moonshot, which is what I would call it. So the people that are trying to figure out how do you just move the nitrogenase genes into plants? There are some labs trying to do that. I personally think that is a overly ambitious goal, but people should be working on it. So I don't want to. I would rather take an organism that already knows how to do it and is living inside the plant and do my manipulations there. Are we the only people doing that? No, we only started working with Gluconacetabacter 6 years ago, and Gluconacetabacter, the Brazilian group that discovered this is several decades ago. I don't know if they're doing the type of manipulations that we're trying to do or trying to do the combinations, but we've overcome quite a few hurdles just in figuring out how to screen these things. And so, let alone how to get them growing in plants so we can really tease out some of these information, but there's another 5 or 6 labs in the US that I pay attention to what they're
And
doing.
you're all focused on NSF for the primary funding,
USDA, NSF, DOE.
but is there a layer of adequate funding across those funding entities? And when Tim asked, in terms of the
And
potential
NSF,
investment dollars, right?
NSF has programs that definitely fit it. Obviously, we got money from USDA on this. That was a first proposal, first fund. It wasn't something we went through multiple tries with that. When we proposed this, USDA loved it. The second time when we proposed it, we were proposing to expand, and they kind of harped on us that we weren't doing enough greenhouse stuff at this stage. I thought greenhouse was getting ahead of ourselves and wanted to do that as part of other grants that were separate. I wanted to still be doing some of the basic science, but so we'll probably try to go back to USDA with some of the greenhouse
So
stuff.
what division in USDA, what programming did it come out of?
What's our program actually named? Now I'm gonna draw a blank.
Yeah, that's fine. I'm just.
It's actually the ecological systems.
I see, yeah.
So, yeah, that was one of the issues we got in our latest round of funding was one of our reviewers tried to tell us we were in the wrong program, even though I checked with the program managers ahead of time and said, are we still in the right program, or is there a better program for us now?
How about DOE and energy?
I have not broken into DOE since our algal work.
I see.
So
Yeah,
DOE's been a hard nut to crack for us,
I guess.
for me anyway. Yeah.
One thing with the crops that you're using, they all have very different root architectures. And I'm wondering what you would envision to be an ideal root architecture for essentially.
Oh, you're asking the wrong person. I know very little about
Well, I
plants.
mean, let's just think in terms of corn. Corn puts down a taproot that goes very, very deep.
Mhm.
You may not know it, but pennycress only goes down about 3 inches.
Yeah,
Has a very shallow taproot and makes a very fine net of root system that's really just below the surface.
our limitation isn't that we wouldn't like to test a lot of them. It's the cost of learning how to deal with each one of those. So yeah, Neil and I both have talked about testing some other plants, but we're working with the budget that's shrinking every day because we're at the end of this. And we're gonna get nice data out of it, and we've got some nice directions, but yeah, I'd love to expand. That's what we proposed in USDA on the second time around, was to test a broader number of plants and see what happens when you
I'm just
go.
thinking about the ecological ecology of the bacteria themselves, where they like to be.
I think there's very little known about it, to be honest with you. In fact, not something I've mentioned, but if anyone else out there has their hands on other native strains of Bubonic Acidobacter or Azotobacter, we'd love to expand beyond the lab strains. In fact, it's something I've been looking at, but sometimes getting those can be thousands of dollars to get them all in hand.
Well, that's what I was going to ask, the material coming from Brazil. Does there need to be another part of the program that's trying to identify these microbes from this environment? Right?
Yeah, so this is what we're proposing to LCCMR this year, is potentially trying to get a better handle on what the microbes are that are already in soils here in Minnesota that might be future beneficial microbes to think about.
Or certain plant associations.
Yes.
For example, in the native prairies, right?
Yeah.
To go out. We still have some pretty large native prairies in southwestern Minnesota that probably you could get permission, and they're controlled by farmers.
Yeah.
Quarter sections, half sections, and in some cases sections
Mhm.
that have not been disturbed ever, right?
Mhm.
So
It
they
feels
would
like there
be,
should be quite a few data sets out there, and people have just gone out to try to look at the, biology of the soils, and what bacteria are associated there.
Yeah, but
I'm not aware of one in Minnesota. I haven't, in terms of that type of
You know,
Do you,
I mean,
I'm just saying,
anything to do something.
We haven't looked at that kind of community analysis ourselves as far as scouring the literature for it. Again, for me, I don't necessarily want to go to ones that are just in the rhizosphere. Really would like to find ones that are endophytes. The endophyte solves so many more of our problems with, all right, now we know it's in. So if we find that's the dominant organism in that plant already, then that's the one we should focus on.
well, if you look at the tall grass prairie. Right, it is a nitrogen limited system. And some of those prairie grasses. Are getting nitrogen someplace, right? That's pretty robust growth, so I'm just thinking. And I thought about it before in terms, as when I learned about the sugar cane.
Mhm.
I tried to put a group together here to think about it, what, 20 damn years ago. Everyone poo-pooed it, right? Everyone, and that's what I was thinking about was. You know, there's sugarcane there, but what here? And I always thought that we ought to look at the native grasses. Because they have that really robust growth in what is considered to be a low nutrient environment. There's a guy that can do it. He can travel.
I'm not doing nothing.
All right, okay, fine. Any other comments, questions online at all? Well, thank you for joining us today. And certainly want to